Welcome back, guys. Welcome back in another antibiotic video lecture from microbiology with Shagni. So in this video tutorial we will going to block the folic acid pathway of bacteria. Okay, so before going to discuss about what kind of antibiotic we will going to use, the first thing that is important to mention, that is what is folic acid pathway. Okay, so let me introduce a new pathway. This pathway bacteria uses to produce or to construct the purine and pyrimidine. Okay, so I hope you all know about what is purine and pyrimidine. So this purine and pyrimidine are the building blocks of the nucleic acid, right? So before DNA synthesis, it is necessary that the bacteria's folic acid pathway will work properly. Okay, otherwise the building blocks will not be there, with the help of which the DNA polymerase will produce the DNA, right? So that is why folic acid pathway is also an important pathway of the bacteria. And I want to mention you that the inhibitors that we will going to use to inhibit folic acid pathway, we will going to discuss about two, or more specifically saying three important drugs. So they all are bacteriostatic. That means what? That means they will not directly ensure the death of the bacteria, but they are reduces their metabolic activity. Okay, so the pathway starts with a reaction between the para-aminobenzoic acid and pteridine in presence of dihydroteroic synthetase. Okay, so now this dihydroteroic synthetase will facilitate the conversion of para-aminobenzoic acid and pteridine into dihydroteroic acid. Okay, now this dihydroteroic acid will convert it into dihydrofolate or dihydrofolic acid. Okay, so now this dihydrofolic acid will be converted into dihydrofolate reductase. Okay, so this dihydrofolate reductase will convert that dihydrofolate or dihydrofolic acid into tetrahydrofolate or tetrahydrofluoric acid, and which ultimately leads to the formation of purine and pyrimidine. Okay, so the first step is the reaction of PABA and pteridine in presence of the enzyme dihydroteroic synthetase, which will give dihydroteroic acid or dihydroteroic acid. So this dihydroteroic acid will convert dihydrofolate or dihydrofolic acid. Okay, so now this dihydrofolic acid will convert it into tetrahydrofolate or tetrahydrofolic acid with the help of dihydrofolate reductase, and which will ultimately leads to the formation of purine or pyrimidine. Okay, so now let's see which kind of drugs available here to inhibit the folic acid pathway. Again, I am mentioning here all will be the bacteriostatic drug. Okay, so the first drug, sulfonamides. It will inhibit the first step of the folic acid pathway, that is the reaction between the PABA and pteridine in presence of dihydroteroic synthetase. Okay, so here these sulfonamides, especially sulfomethoxazole, this for example, they are acting as a substrate analog, because as it is a enzyme, it will react with its substrate, right? That is para-aminobenzoic acid and pteridine. So here these sulfonamides will act as a substrate analog, and as a result they, that is dihydroteroic synthetase, cannot convert the para-aminobenzoic acid and pteridine into dihydroteroic acid. Okay, so it is again a bacteriostatic activity. Okay, and this kind of inhibition is known as competitive inhibition. Okay, that means if the concentration of the para-aminobenzoic acid and pteridine increases, then the inhibition of the sulfonamide may be decreased. Okay, so now this dihydroteroic acid will convert dihydrofolate or dihydrofolic acid. So now this dihydrofolic acid will be converted into tetrahydrofolate with the help of an enzyme called dihydrofolate reductase. Okay, so this enzyme will be inhibited by trimethoprim, which is which is used to treat the infection caused by gram-negative bacteria, for example Enterobacteriaceae. Okay, and I would like to mention you that this sulfonamide will use to treat nocardiasis, bacterial UTI infection, along with it will use to treat toxoplasmosis, that is various kind of parasitic infection caused by cats. Okay, now it will ultimately lead to the formation of purine and pyrimidine, although the amount will be lowered drastically because here we have used various kind of various kind of drug to inhibit the pathway. Also in some treatment, especially for pneumocystis carini infection, which is a kind of yeast infection, we can use both the sulfomethoxazole and trimethoprim. Okay, that means we will block the first step, along with we will block the third step. Okay, so this mixture is known as Bactrim. Okay, so it will prevent the pneumocystis carini infection caused by various kind of yeast. So it is a immunocompromised, this pneumocystis carini infection is predominant or mostly occur to immunocompromised person. For example, a person who are suffering with AIDS are at high risk of those kind of pneumocystis carini infection, and under such circumstances we will use sulfomethoxazole and trimethoprim. Okay, that is collectively known as Bactrim. So let's try to revise about the folic acid pathway. So folic acid pathway all starts with the reaction of PABA and pteridine. Okay, PABA means para-aminobenzoic acid. Okay, so they will be converted into dihydropteroic acid with the help of an enzyme called dihydropteroic synthetase. Okay, so this dihydropteroic synthetase will block the sulfonamides. So sulfonamides is a kind of sulfomethoxazole. Okay, that means sulfonamides, example of sulfonamide is sulfomethoxazole. So this sulfomethoxazole act as a substrate analog. Okay, and prevent or inhibit the dihydropteroic synthetase competitively, which is known as competitive inhibition. Okay, so now dihydropteroic acid will be converted into dihydrofolate or folic acid in the subsequent step, and this dihydrofolic acid will be then converted into tetrahydrofolate or tetrahydrofluoric acid with the help of dihydrofluorate reductase. So now this dihydrofolate reductase will be inhibited by trimethoprim. Okay, so it will be we are using trimethoprim for treating gram-negative Enterobacteria infection. Okay, and from the tetrahydrofluorate we will ultimately get purine and pyrimidine, which the DNA polymerase will use for DNA synthesis. Okay, so here we are using three type of drug. Try to remember again. One is sulfonamide. Okay, for example sulfomethoxazole. This will competitively inhibit the dihydropteroic synthetase. Okay, and prevents the formation of dihydropteroic acid. Okay, by acting as a substrate analog. And the second one is inhibition of the dihydrofolate reductase. Okay, so this dihydrofolate reductase inhibition is done by trimethoprim. Okay, which especially prevents Enterobacteriaceae gram-negative infection. Okay, now this trimethoprim and sulfonamide, for example sulfomethoxazole, can also be collectively applied. So such kind of drug is known as Bactrim. So Bactrim have some special properties. It have the ability to destroy pneumocystis carini infection, which is a kind of yeast infection. Okay, so remember this pneumocystis carini infection, which is a yeast infection, is predominant or person who are immunocompromised. Okay, that means person who are, let's say, suffering with AIDS are at risk in those pneumocystis carini infection. Okay, so that was all about folic acid pathway inhibitors. So I hope this video will be helpful to you, and thank you for listening to this class.